and today I'll be teaching you about central dogma. Central dogma is basically how our DNA is read and later transcribed into RNA and finally translated into proteins. So how our DNA is read and finally expressing into proteins requires two steps, transcription and translation. The main purpose of transcription is to change DNA into RNA. Before I start explaining transcription, you have to remember the key difference between DNA and RNA. In DNA, adenine pairs with thymine, while in RNA, adenine pairs with uracil. The whole process of transcription is happening in the nucleus. This is an enlargement of what is happening in here. There are three main processes in transcription: initiation, elongation, and termination. During initiation, an enzyme called RNA polymerase will attach itself to the promoter region of a DNA strand. The promoter region encodes for the start of the gene. After the RNA polymerase has attached itself onto the promoter, it will start the elongation process and start transcribing the DNA. The RNA polymerase will add complementary bases onto the new DNA strand. It will pair adenine with uracil, cytosine with guanine. The new RNA strand is called the mRNA, or messenger RNA. It is basically the message transcribed from the DNA. The mRNA will elongate in a 5' to 3' direction while reading the DNA in a 3' to 5' direction, as it will always read it in an anti-parallel direction. So when does the RNA stop transcribing the DNA? Well, eventually, the mRNA will come to a termination sequence on the DNA strand. The termination sequence tells the enzyme to stop transcribing and end the elongation process. After the mRNA has stopped transcribing the DNA, it will leave the nucleus and enter into the cytoplasm. The translation process starts when the mRNA has to be translated into individual amino acids. The amino acids will then link together to form long chains of proteins. The translation process starts when the mRNA enters into the ribosomal RNA, also known as the rRNA. And then the tRNA, also known as the transfer RNA, will read the mRNA's message in triplets, also known as codons. This means that it will read the mRNA message 3 bases at a time. Before we go any further in learning about the translation process, let's first learn about how the codons are read from this RNA codon chart. So when do the proteins know when to start the translation process? Well, there's a start codon which initiates the translation process. The start codon is AUG, which codes for methionine. All proteins start with the start codon. Well, there are 3 stop codons. The stop codons tells the enzyme when to stop the translation process. The 3 stop codons include UAA, UAG, and UGA. In this example, the UGG anticodon is a complementary base pair to the ACC on the mRNA strand. The tRNA will bring the anticodon and the amino acid onto the ribosomal RNA. When it arrives on the ribosomal RNA, it will then deposit its amino acid, which will then be added onto the growing peptide chain. After it has done that, it will leave the rRNA, and a new tRNA will then come in and deposit its own amino acid. Once the stop codon is read, the translation process ends, and then the growing peptide chain will then be cleaved off, and then it will become a whole new chain of protein. Proteins account for our genes, which include our hair color and our eye color. And that is the whole process of transcription and translation.